Robert's daughter brought something different to Sunday lunch this time. Not the kefir she'd explained in our second episode. Not the dynamometer she'd pulled from her bag 3 months ago and placed between the main course and dessert. Something smaller. A home collection kit, a small lancet, a blood collection card, a prepaid return envelope, and a barcode she explained he would use to retrieve results from a secure online portal. "This one actually measures your brain?" Robert asked, looking at the box. "It measures how fast you're aging," she said, which turns out to be the same thing. In our last video, Robert's daughter described grip strength as the proxy measure she used in research, a signal that could estimate cognitive risk across populations too large to test individually. She said she'd never expected to use it at her father's kitchen table. This video is about the direct measure she brought next. I'm Dr. Ben, and in this video I want to tell you about something that has existed for a decade in research settings, that has predicted cognitive decline more precisely than almost any other biomarker we've discussed in this series, and that, as of the last 18 months, has crossed the line from laboratory science into something you can actually order from home. It's called an epigenetic clock, and specifically, the one that has shown the strongest and most consistent evidence for predicting cognitive aging, DunedinPACE. Let me start with the concept, because it requires understanding something about your DNA that most people learned in school but probably haven't thought about since. Your DNA sequence, the genetic code you inherited, is essentially fixed at birth. What is not fixed is the pattern of chemical tags that sit on top of that sequence, instructing which genes are expressed and which are silenced. These chemical tags are called methyl groups. The field that studies them is called epigenetics, literally, above the genetics. And here is what researchers discovered, starting in 2013 with work by Dr. Steve Horvath at UCLA. The pattern of methylation across specific sites in your DNA changes in a highly predictable way as you age. The changes happen at different rates in different people, which is why two people of the same chronological age can have very different methylation signatures. And the difference between your methylation predicted biological age and your chronological age, the gap between how old your DNA says you are and how old you actually are, turns out to be one of the most powerful predictors of morbidity and mortality that exists. This is what an epigenetic clock measures. Not how many years you've been alive, how many years your biology has been operating at. Now there are several generations of epigenetic clocks, and they measure different things. The first generation, including the Horvath clock, measured biological age. A second generation, including Grimmage, calibrated that measurement specifically against mortality risk and plasma protein signatures, making it a significantly stronger predictor of health outcomes. Grimmage, which Robert's daughter uses in her research, has shown in multiple large cohort studies to outperform other epigenetic clocks in the prediction of age-related clinical phenotypes and all-cause mortality. But the clock that matters most specifically for this channel and for the cognitive aging question is DunedinPACE. DunedinPACE is different from Grimmage in a specific and important way. Grimmage measures how much accumulated damage your biology has already experienced your biological age relative to your chronological age. DunedinPACE measures something different, the current speed at which you are aging right now. Not how old your biology is, how fast it is moving. The difference matters, because DunedinPACE can change in response to intervention. A score of 1. 0 means you are aging at exactly the average rate for your cohort. A score of 1.2 means you are aging 20% faster. A score of 0.85 means you are aging 15% slower. And because it measures pace rather than cumulative damage, it is sensitive to changes, which means it can actually detect whether the things you are doing to protect your brain are working. In the Framingham Heart Study Offspring Cohort, 2,296 adults, average age 61, followed over 20 years faster DunedinPACE, was robustly associated with worse global cognition and steeper decline in cognition over time, independent of education, sex, age, and smoking status. Published in November 2024 in Alzheimer's and Dementia. A separate paper, published in the same journal in July 2025, examined the metabolomic signatures associated with faster DunedinPACE. and found that the pathways driving accelerated biological aging overlapped substantially with the systems we have covered throughout this series. Inflammation, gut microbiome disruption, circadian misalignment, and impaired glucose regulation. The aging clock, in other words, is measuring the cumulative effect of the same processes this series has spent 19 episodes discussing. And a 2024 study tested DunedinPACE against brain structure in 3 independent cohorts spanning midlife to advanced age, including the Alzheimer's Disease Neuroimaging Initiative, and found that faster DunedinPACE was associated with smaller brain volume, reduced cortical thickness, and reduced hippocampal volume. Not just cognitive test performance. Structure, now the access question. Because research findings are only half of what makes a biomarker clinically useful, DunedinPACE is available from at least one consumer testing service, True Diagnostic, as a direct-to-consumer blood test requiring no prescription and no clinic visit. A lancet is used to collect a small blood sample at home, which is returned in a prepaid envelope. Results, including DunedinPACE score and a breakdown of major epigenetic aging metrics, are delivered through an online portal. Pricing as of 2025 ranges from approximately $200 to $500, depending on the panel depth. I want to be specific about what this is and what it isn't. It is a research-grade biomarker with strong evidence in large longitudinal cohorts for predicting cognitive and health outcomes. It is sensitive to lifestyle change, meaning it can be used to track whether interventions are having a measurable effect on your rate of biological aging. It is not a diagnostic test. It is not an FDA-cleared clinical test for any specific condition. It should be used as exploratory information, ideally discussed with a physician or health professional who understands its context. And the interpretation of a single DunedinPACE score requires understanding reference ranges, cohort norms, and individual variability, which a clinical conversation provides. Robert's daughter offered all of this to him over lunch, in the same systematic way she'd explained the gut microbiome to him years earlier. He said, "So you're telling me you can now measure how fast I'm aging?" She said, "We can measure how fast your Dina is aging," which correlates strongly with everything else. He said, "And can it change?" She said, "That's the most important question you've asked me at Sunday lunch in 3 years." Because it can. A longitudinal multi-cohort study published in eBiomedicine in December 2025 found that specific lifestyle factors move DunedinPACE, some accelerating it, some slowing it. Robert's daughter walked him through the list at the table, and he recognized each item as something this channel has already covered. Smoking accelerates DunedinPACE, the single largest modifiable factor. Higher BMI, elevated glucose levels, and poor blood pressure control also accelerate it. Conversely, physical activity and a healthier diet slow it. In order of effect size, smoking cessation, glucose regulation, blood pressure management, BMI reduction, increased physical activity, and dietary improvement. Physical activity is the morning walk that grew Gerald's hippocampus. Glucose regulation is the dinner timing and fasting insulin we covered in the chrononutrition videos. Blood pressure management is the cardiometabolic component of US Pointer. Dietary improvement is the Mind diet, the fermented foods, the pomegranate seeds. And smoking cessation, for those it applies to, is the strongest single factor, the one that moves the number the most, in the direction of longer biological life. This is what the series has been building toward. Not a supplement, not a drug, not a single intervention. A measurable, quantifiable reduction in the speed of aging itself visible at the cellular level in your Dina methylation pattern produced by the accumulated effect of the specific systems this channel has spent 19 episodes explaining. Robert's test results came back 17 days later. His DunedinPACE score was 0.97. Slightly below average he was aging, biologically, at 97% of the pace of his age cohort. Not dramatically slow, but not fast. His daughter was quiet when she saw the number. He asked her what she was thinking. She said, 3 years ago I would have expected something closer to 1.05 or 1.08, based on your profile at the time. The kefir, The walk, the dinner timing, the grip work, I think it's showing up. Robert said, "You can't know that." She said, "No, I can't prove it. But I can tell you that 0.97 is a better number than I expected to see. And we're going to test it again in a year." Here is the question to bring to your physician, or to begin exploring independently. I've been reading about epigenetic clocks, specifically DunedinPACE, as a biomarker for the pace of biological aging and its association with cognitive outcomes. Is this something you'd recommend exploring given my health profile? And if I pursue it independently through a consumer testing service, what would be most useful to know when interpreting the results? That question opens a conversation about a biomarker that most physicians are beginning to encounter, but that many have not yet integrated into routine clinical discussion. A physician who follows preventive medicine or longevity medicine will engage with it directly. One who hasn't yet encountered it will be willing to learn alongside you, because the research is now published in JAMA-level journals, and the question is a legitimate clinical one. And if consumer epigenetic testing isn't where you are right now either, because the cost puts it out of reach, or because you'd rather wait for clinical integration, the most important thing this video can leave you with is this. The interventions that most reliably lower DunedinPACE are the same ones this series has covered across 19 episodes. They are free. They require no test kit and no blood draw. The morning walk, the dinner timing, the fermented foods, the medication review, the homocysteine check, these are the factors the eBiomedicine data identifies as the ones that actually move the speedometer. The test is useful for tracking. The interventions are the point. This is where this channel has been going. Not toward alarm, not toward a list of things to stop doing, toward the point where the interventions in this series produce a measurable, visible, quantifiable signal that biological aging is responding to what you're doing. A test. A number. A year from now, another number. The same thing Robert's daughter has been doing at Sunday lunch for 3 years, with a dynamometer and a kefir recommendation and a home blood collection kit. Measuring so that you know. I'm Dr. Ben. Thank you for staying with this series. Subscribe for more, because the research is still moving, and so is this channel. The information in this video is for educational purposes only and does not constitute medical advice. Epigenetic testing is not a diagnostic test and should not be used to self-diagnose any medical condition. Always consult your doctor or a qualified healthcare professional before making changes to your health management plan based on epigenetic testing results.